Hello students, welcome back to a new video of Sunanda's tutorial. In this video, I will discuss about the gluconeogenesis, and it is the last part of the lipid metabolism. So let's discuss what is gluconeogenesis. It is the process that allows the body to form glucose from non-hexose precursors or non-glucose products, particularly from glycerol, lactate, pyruvate, propionate, glucogenic amino acids. So from these products, glucose will be formed. It is the synthesis of new glucose from non-carbohydrate precursor, providing glucose when dietary intake is lacking. When there is less intake of glucose or the amount of glucose is decreased in our body, then this gluconeogenesis pathway is followed inside our bodies. Then the gluconeogenesis is the building of a novel glucose molecule in the body as compared to glucose, which is split from prolonged storage molecular glycogen. It is primarily occur in the liver. That means the gluconeogenesis process occur inside the liver. But it can also occur in minor level in small intestine as well as in kidney. It is more or less reversal process of glycolysis, because in case of glycolysis, glucose breakdown to form a non-carbohydrate product, that is pyruvate. But in case of gluconeogenesis, from that pyruvate, glucose is formed. So it is approximately a reversible process of glycolysis. Then the pathway of gluconeogenesis. Gluconeogenesis originates in liver or in kidneys cytoplasm or inside the mitochondria. To make oxaloacetate, at first oxaloacetate will be formed. It requires two pyruvate molecules. Then this require one ATP molecule also. The first reaction, where the pyruvate is converted to oxaloacetate in the presence of pyruvate carboxylase. Then oxaloacetate is converted to malate, which then transported out from the mitochondria. Once malate leaves the mitochondria, it is oxidized back to oxaloacetate. The enzyme phosphoenol pyruvate carboxykinase convert that oxaloacetate to phosphoenol pyruvate. So the first reaction, where the pyruvate is converted to oxaloacetate by the enzyme pyruvate carboxylase. Then NADH converts this oxaloacetate to malate, and this malate transported out of the mitochondria, and once the malate leaves the mitochondria, it again oxidized to form oxaloacetate. Then from the oxaloacetate, phosphoenol pyruvate or PP will be formed by the enzyme phosphoenol pyruvate carboxykinase. By reversing glycolytic process, phosphoenol pyruvate is converted to fructose 1,6-biphosphate. That means the rest of the process till the fructose 1,6-biphosphate is just the reverse of the glycolysis. That means first phosphoenol pyruvate will be converted into 2-phosphoglycerate, then 2-phosphoglycerate to 3-phosphoglycerate, 3-phosphoglycerate to 1,3-biphosphoglycerate, then glyceraldehyde 3-phosphate will be formed. A reversible reaction occur between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These two again converted to fructose 1,6-biphosphate. then the enzyme, then the fructose 6 phosphate will also be formed. Then phosphoglucoisomerase convert fructose 6 phosphate to glucose 6 phosphate. Glucose 6 phosphate generates inorganic phosphate that yields free glucose. That means phosphate group will be removed and only glucose is obtained, which enter the blood. And glucose 6 phosphatase is the enzyme which is involved in the last step, that is conversion of glucose 6 phosphate to simple glucose. So let's revise the whole process. At first, from pyruvate, oxaloacetate is formed by the enzyme pyruvate carboxylase. Then from oxaloacetate, phosphoenol pyruvate will be formed by the enzyme PEP carboxykinase. Then from phosphoenol pyruvate, 2-phosphoglycerate will be formed. Then from 2-phosphoglycerate, 3-phosphoglycerate will be formed. Then from 3-phosphoglycerate, 1,3-biphosphoglycerate will be formed. Then from 1,3-biphosphoglycerate, glyceraldehyde 3-phosphate will be formed. Then from that glyceraldehyde 3-phosphate, then from glyceraldehyde 3-phosphate, fructose 1,6-biphosphate will be formed by the enzyme aldolase. Here in gluconeogenesis, by the help of the enzyme aldolase, glyceraldehyde 3-phosphate converted to glucose 1,6-biphosphate. Then from fructose 1,6-biphosphate, fructose 6-phosphate will be formed by the enzyme fructose 1,6-bisphosphatase. Fructose bisphosphatase. Then from fructose 6-phosphate, glucose 6-phosphate will be formed. Then from glucose 6-phosphate, glucose will be formed by the enzyme glucose 6-phosphatase, which is the main enzyme which is required in the last step. And here the phosphate group will be removed as inorganic phosphate, forming the glucose. This glucose ultimately enters inside the blood. So the first step, in which the pyruvate by consumption of 1 ATP convert into the oxaloacetic or oxaloacetic acid, then oxaloacetate by NADH convert to malate. The conversion to malate enables the molecule to transfer out of the mitochondria, then mitochondria transfer out of the malate transfer out of the mitochondria and converted back to oxaloacetate in the cytoplasm. Then in cytoplasm, malate converted to oxaloacetate, then oxaloacetate by consuming 1 guanosine triphosphate converted to phosphoenol pyruvate. It then passes through same intermediate that glycolysis does. The endoplasmic reticulum is the location of final reaction. Then in endoplasmic reticulum, the glucose 6-phosphate, from glucose 6-phosphate, glucose will be formed by the enzyme glucose 6-phosphatase. Then the glucose is transported out of the cell into extracellular environment by glucose transporter. So it is the overall process or pathway of gluconeogenesis. Now what is the importance of gluconeogenesis? During deprivation, gluconeogenesis cycle is important for blood glucose regulation. When the glucose level is low in the blood, then the gluconeogenesis cycle or pathway is followed. Many cell or tissue including RBC or red blood cell, neuron, skeletal muscle, the medulla of kidney, testis, embryonic tissue rely on glucose to meet their energy needs. The neoglucogenesis or gluconeogenesis, both are same, cycle removes metabolites such as lactate and glycerol from bloodstream. So it is all about the importance of gluconeogenesis. During deprivation, it is important to maintain the glucose level of the blood. Then many cells rely on the glucose to meet their energy needs. So glucose have to prepare inside the body. So gluconeogenesis pathway is followed, and this cycle is very important as it removes many metabolites, that is lactate and glycerol, which are not so good for our health. So this pathway is very important. So it is all about gluconeogenesis and its importance. So here with this video, the complete lipid metabolism chapter is finished. In my next video, I will discuss some other important topic of biology. So stay tuned and enjoy my videos like this. Thank you.